a significant thing, and it is. It's actually quite significant having evolutionary loss. And then, so we saw how the snake lost its forelimbs by eliminating the expression domain required for its forelimbs by moving these Hox genes up to here. And the hind limbs, we just discussed briefly, were eliminated by interrupting signaling mechanisms that lead to the formation of the hind limb. Looking at the, one of the sort of striking things when you start looking at embryos and doing what we would call, say, comparative embryology, is the inherent similarities that related organisms have at the embryonic stage, or during embryonic development. You can see an example of this here. These are several different vertebrate embryos at similar stages of development, and you can see the similarities between them all. Right? They all have that same curvature, the head, the eye, the pharyngeal arches are present in all of these guys, and they're all kind of the same looking. This is just another picture of the same thing. You can see comparing a chicken and a human embryo, you can see how similar they look. The human embryo is obviously turned around, but you can sort of see all of the similarities there. There's the big head up here that curls around and comes down to the tail. The big head that curls around and comes down, and then the tail kind of comes across like that. There's the heart in both cases. Right? So there's a lot of similarities in terms of how things look. On the other side is a series of pictures of embryos. Not the best series of pictures, but chicken, human, mouse, and then a turtle. And really, especially the chicken, human, and mouse all look really, really similar to one another. The turtle has a different looking head. But there are a lot of structural similarities that you see. Things like pharyngeal arches that we have as embryos but not as adults, or the tail and things like that, all those kinds of similarities. So the idea is kind of that we all start pretty much looking the same, and then we diverge as we start using those toolkits in different ways. So we end up with starting off with kind of the same body plan that's very, very similar, and then use those toolkit genes in different ways to diverge and form different anatomical features. Right? So that's one way of looking at it. And if you look at it, this is looking at neural tube formation in amphibians and also in fish. Right? And you can see that when we look at this process, as we've done on a number of occasions, it's fairly similar. Right? There are some tweaks some things that are a little bit different, but the whole process is fairly similar from one species to another. So even mechanistically, things look pretty similar from one species to another. So something to consider as we look at this kind of stuff. And then, this is the kind of one of the ways that we can think about using those toolkit genes in different ways to generate diversity. Looking at homologous structures here, the four limbs of vertebrates. Of course, we see the arm, the bat of a wing, the pterodactyl wing, the whale's front flipper, the pectoral fin, and then a horse's leg. And focusing mainly on the hand region, what we see here is, remember, when we looked at limb development, we saw those three phases of Hox expression. The first phase leads to the formation of the humerus, second phase, ulna and radius, and then the third phase causes a turn in that axis. It starts off, it comes along here, and then it causes a turn in that axis. So it kind of goes horizontal, and then it flips vertical, and that's what gives us our hand structure, and basically the structure past the wrist. When you look in fish, they don't have that third phase, so they don't have a hand or anything past the forearm. Right? They just have nothing here. But what we're looking at here is the divergence of that hand. Right? Now, mainly when we look at these limbs, it's hard to, this diagram doesn't show it, at least in the case of the human and the horse, but everything else is pretty much the same. Right? The forearm and the upper arm, or foreleg and upper leg, or whatever the appendage is, looks pretty much the same. Right? So we have the humerus, ulna, radius, same thing in the back. Right? Everybody has that stuff. It's really the big differences are in proportion. So the proportions of those bones, the relative sizes of those bones differ. But really where you see the difference is down in the hand region, the wrist and the hand. And that's where we have that's the divergence of these different structures. And of course, we know what happens with our hand. And the limbs of, say, like a bat, that just has basically really, really long fingers. The bat, you can see the arm is kind of extended. There's not much of a wrist, but there's really, really long fingers in the wing of the bat. And it's sort of a similar, well, not similar, but somewhat of a similarity there with the whale, where we have femurs, ulnar radius. These guys are shorter, and then we have kind of really long fingers as well, but they're bunched together to make this pectoral fin, right? And this is probably the pectoral fin of a humpback whale, which has an especially long pectoral fin. Other whales are going to be a little shorter, so it'd be maybe like back to there. But these are the digits here. You can see the 5 digits, so they just have really long fingers. And then in something like a horse, an animal with a hoof, basically we have the wrist up here, and then this is the hand, and then the fingers, and then the hoof is basically a modified fingernail. So the horses are kind of like walking around on one finger in a sense. It's obviously modified so it can do that well, but that's kind of how they're walking along. But all of this is modifying that last step in the process. The earlier steps happen in a very similar way from organism to organism. It's that last step that you can tweak to generate different types of structures. So kind of a cool way of doing it. All right, so I'd like to end by talking about a couple of other examples of studies in Evo Devo where people have looked at how evolution has evolved to generate differences in organisms. And what we're looking at here are some fish, obviously, but there are 2 different types of fish. There's these guys here and this one here. These guys are sort of normal fish that you might find living in freshwater, bodies of fresh water. Right? They're little fish. They have nice eyes. They see quite well. These guys are very, very close relatives, but they don't live on the surface. They live in caves where it's completely dark. All right? Well, if it's completely dark in a cave, you don't need eyes, right? Even if you had eyes, you couldn't see anyway because there's no light. So why do you need eyes? Well, so what happened is these cave fish lost their eyes. Right? So they no longer have eyes. Well, and you can see close-up pictures here. Here's the surface fish. Here's the cave fish. And you'll notice also by looking at them that the pigmentation's different as well. The cave fish are basically devoid of color, while the surface fish have some coloration to them. Right? Once again, why do you need pigmentation if you are living in a place where there's no light and you can't see, nor can anybody else? So it's a nice example of natural selection and adaptation, right? So one of the ways that we can think about this is, think about natural selection is, we normally talk about it as selecting for favorable attributes, but it also will select against unfavorable characteristics. And if you're a surface fish and you need your eyesight, any kind of change that causes a fish to lose its eyesight is going to be detrimental to it, right? So having eyesight is going to be selected for, lack of eyesight is going to be selected against in surface fish. But in the cave fish, there's no selection against lacking eyesight, right? If you lose your eyesight in the cave fish, well, who cares? You can't see anyway, so losing your eyesight makes no difference. All right. So one of the things we can sort of think about and look at is, well, how did that happen? Right? How did it happen that cave fish lost their eyes? Right? It was almost certainly because of some sort of a change occurred that caused the fish to lose their eyesight, and because it wasn't a liability, in fact, it might have even been an asset. It ended up persisting in the population and becoming the norm, right? Taking over the population. So what happened there? Why did it happen? Well, in other words, and perhaps a different question would be, why is it beneficial? Why is it beneficial to the fish not to have eyes? Right? Well, that, nobody really knows the sort of definitive answer for that question. But one sort of real likely possibility is one of basically energy, right? Think about what it takes to make an eye, and then maintain an eye. To make an eye, costs energy. Eyes are very active, physiologically active organs. They're taking in light, and that light, sunlight, is converted into a chemical impulse that's sent to your brain, and the brain processes it. All that takes energy. All right? So, losing the eye might actually not be a bad thing as long as, if you're not using your eyesight anyway, that's sort of wasted energy. You have more energy to use on other things if you don't have to worry about making and maintaining an eye. All right? And also, if you are in a cave and you're swimming around blind, literally, there's greater chance that you're going to be running into things, and that could damage your eye, and that damage could cause injuries and infections and things like that that could also be a liability. So there's probably actually an advantage to cavefish not being able to see. Well, whatever the evolutionary reasons behind it, one of the things that is certainly, or one of the things that researchers have done is tried to figure out, how did it happen? How did the cavefish lose their eyes? What's the sort of molecular mechanism behind eye loss? And they actually did some experiments with this. And they've done a number of different types of experiments. And one of the first types of experiments they did was a transplantation experiment. They took fish embryos and they swapped lenses from the eye. So they took a surface fish lens and put it in a cavefish, and a cavefish lens and put it in a surface fish. And the nice thing about fish is that, they have two eyes, of course, right? So you could do the transplant on one side and leave the other side alone. And that will tell you, so you have an internal control. You have a built-in control in this. And when they did the swap, they swapped phenotypes, right? So if you put a surface fish lens in a cavefish, the cavefish forms an eye. And by all accounts, it's a functional eye. If you do the reverse, if you put a cavefish lens into a surface fish eye, they have no eye on that side. The eye degenerates. So what actually happens developmentally in the cavefish is it actually starts to make an eye. And then once it starts forming, it forms this small kind of abnormal lens, and it just sort of gets messed up and the eye doesn't form. So it starts to form an eye, it tries to form an eye, but it's unsuccessful. And the eye eventually just, kind of goes away. So one of the things that people noticed was that hedgehog signaling seemed to be important here. Wow, hedgehog again, right? How many times have we talked about hedgehog? Here's one more. And what we're looking at here is basically a situation where they injected hedgehog mRNA into one side of the surface fish embryo, but not into the other side. So once again, we have our internal control here. Right? And basically, if you're looking at the top, what you're looking at is a micrograph showing the brain and early development of the eye optic vesicles of the fish. And the right-hand side is the untreated side. The left-hand side is the hedgehog injected side. And you'll notice a lot more, a lot greater level of development on the right than on the left. Right? That's because the hedgehog signaling is blocking eye development, blocking eye formation. So when you look a little bit later on, here's the normal eye with the lens in it. Here's this sort of stunted eye with this smaller lens in it. And when you get to the the adult fish being formed, this is what you get. So this is a surface fish. You can see the pigmentation here, right? And what do you get? No eye. Right? The fish has no eye on that side. It has a perfectly normal eye on the other side. In the case of the cavefish, if you put the surface fish lens in the cavefish eye, you see an eye that's approaching normal, right? So it's kind of an interesting scenario that what's happening here is that hedgehog signaling is regulating eye development, and overexpression of hedgehog is blocking eye development in the cavefish, right? So kind of a nice little system to examine this process. So people have done all sorts of different experiments with these kinds of things. It's really a fun little story to try to explain how this thing happened. And it's all getting back to looking at development. How does the eye develop? what goes wrong with eye development in the cave fish that goes right in the surface fish, and that leads us to this whole sort of scenario.
Did you say there was too much hedgehog in the cave fish?
Yeah, when you express hedgehog in the eye field, the eye starts to degenerate. It doesn't develop properly, and it eventually degenerates. But it's because of crosstalk between the lens and the rest of the eye. It messes up that signaling between the lens and the rest of the eye, and the eye doesn't develop properly. That's kind of a fun little system. So that's kind of an interesting thing to look at. Here's another one. Fix that. This is looking at a group of fish called stickleback fish. Okay? And these stickleback fish come in a number of different varieties, but it turns out that long ago, after an ice age, these stickleback fish became separated between different environments. And some fish, like the one on the top, found themselves living in open water, deeper water environments, right? Whereas the ones on the bottom found themselves living in very shallow water environments, all right? Because of, basically what happened is the ice age went away. Some fish got trapped in lakes that were very shallow. Others got trapped in lakes that were deep. And basically what happened is they encountered different environments, right? More specifically, they encountered different predators, right? There are different predators in those different environments. So let's look at the fish on the top. Okay, this is the open water fish. And the red that you see here is highlighting bony structures. Right? And what we want to focus on are a couple of things. These here, those are spines, right? And then you see all this kind of thick red stuff here? That is basically bony plates that form on the outside of the fish. It's like almost like a little bit of armor, right? So if you were to look at this fish head on, what you would see would be something like this, right? You would see the fish coming at you with a spike standing up in the air and then two spikes sticking down at the bottom, all right? Now, if you're a predator, a larger fish that's trying to eat this little fish, and you see this thing with spikes sticking off of it, you're going to think twice before you chomp down on that, right? It's going to hurt. And you can get damaged, injured, and stuff like that, right? So that armor in those spines are very, very good protection against large predators like fish, all right? So if you have all this armor, you're going to be protected against fish. But look at this guy on the bottom. What do you see here? No armor plating, no big spines along the belly, a couple of smaller spines along the back, right? Now this guy, you'll look at him, and what are you going to see here? You'll see something like that. Which one are you going to eat? Right? You're going to eat this guy if you're a bigger fish, right? So these guys have all the armor plating because of their predator defense. These were the originals, right? These were how the stickleback fish all used to look, right? But what about these guys that got trapped in the shallow water? Well, their predators are no longer large fish. The water's too shallow. Large fish don't live there, so there's no predation. But here, so once again, they don't need all the armor plating. Okay? So once again, it's an energy savings. You don't make stuff you don't need. But it turns out that actually the spines are a liability in the shallow water. The predators of these fish are insect larvae. And some of those insect larvae, now these aren't big fish, right? These are, 1.5 inches long. These are small fish. If you've ever seen insect larvae, some of those insect larvae, they can be big. They can be good-sized things, especially some of them that live in the water. They can be quite large. I've seen them, like that big. Right? And they have these little, claws and things. And what they'll do is they will grab onto the spines. The spines are like little handholds for them. There's a fish going by, gotcha, lunch, right? They eat it because they've trapped it, right? So the spines are actually a liability, right? The spines are a liability. They lose the spines, lose the armor, because that is actually worse for them than having them. So what's protective in one situation is actually, the reverse, the opposite. It's actually more dangerous in another situation. So you can see how that change took place. And basically, without getting into all the details here, The short answer to all of this is that most of the so most of the change in phenotype here between the two is actually dependent upon a single genetic change. Right? It's mostly dependent upon a single genetic change. So a single gene is changed, and that leads especially to the reduction in the spines. That's a really big, reduction here. So have you guys done some of your advanced genetics, right? You've done like what's the term is running out of my head right now, the log scores, and the thing where you can, what's the name for it? Where you can, in a multifactorial trait, you can figure out which percentage of the trait.
No, that's human genetics.
What?
That's human genetics.
That's human genetics. Oh, okay. You did it in human genetics, not. Okay.
You want to take that?
Hmm?
You want to take it?
I can't remember what it is, but there's a way of, let's say you have a phenotype. You don't remember what it is? No, you don't remember. There's a, it's like multifactorial analysis or something like that. It's a way that you can say, all right, let's say you have 5 genes controlling a single trait. You can figure out, what the percentages are. In other words, gene number 1 controls 30%, 2 controls 80%, or that's too much, but 60%, and then the other 3 split up the remainders. So when you talk about, polygenic traits, if you have 5 genes, it doesn't mean all 5 genes contribute equally. And by doing this sort of thing, you can figure out what it is. And it turns out that the bulk of the genetics shows that a single genetic change accounts for the majority of the phenotypic changes in these fish. So it's kind of a fun little thing to examine how this all occurs. And for those of you that will still be here next spring, I actually teach a course called Evolution of Developmental Mechanisms, or EVO, where this is the kind of stuff we do all semester. We look at, in more detail at the stickleback fish. We look at the blind cave fish. We look at changes in Hox expression and how that leads to changes in phenotype. We look at butterfly eye spots. A lot of these things that I've given you, 3-minute little summaries of, we'll spend, in some cases a week or so or more on each of these, and really look at them, look at how they work. So if you're interested and around, that'll be next spring. So I think that's it, right? That's the last slide.
Yep.
Yeah, so that's it. We can use whatever time we have left, if you'd like, to do any reviewing for the final. It's just 3 chapters. It's just the 4th exam. There's no cumulative stuff on there. It's just another exam. There's nothing, kind of unique or anything about it. I have the problem set up on Angel, so that's available to you for sample types of questions, and I can certainly answer anything you want. But no surprises. Same types of questions, same types of formats and things like that you've seen 3 times already before. Right? Here, of course, you're going to want to be familiar with what the different types of genes do. So if we're looking at, I think the first chapter is fly development. So you want to know the hierarchy of the genes and what they all do, all those gene interactions and stuff like that. You want to be aware of all that stuff. Know how the gene regulatory interactions take place between the maternal gap and parallel genes. And then, you should know about the signal transduction cycle, that crosstalk with the segment polarity genes with hedgehog and engrailed and all that kind of stuff. Right? So you should know all that whole cycle, the sort of reciprocal interaction between those cells, and of course the homeotics and how they work to specify segmental identity. And then, after that we talked about in chapter 16, the first part was talking about basically how the organizer works in the frog. So you should know about all the genes involved there and how we set up the nucleus center where we have that nuclear beta-catenin and TGF-beta overlap. That turns on the CMLOD gene, which then signals the organizer to make all of its stuff. And the function of the organizer genes is largely to inhibit the BMP signaling, and that allows for dorsal development to take place. Right, so that's the frog. And then, of course, the rest of that chapter was all about the vertebrates' Hox genes and looking at the nested expression patterns and how those expression patterns correlate with the setup of the embryo. I'm not going to ask you specific questions like which Hox gene defines the border between this and that. More general sorts of questions about those sorts of things. Big part of that chapter, though, was limb development. So you should know how limb development works. Know how all the different limb axes are specified, and about the three Hox phases and how they specify the positional information in the limb. Then, of course, this last chapter, the Evo-Devo chapter, we kind of just went over that. So, looking at the conservation, and the key here is really altering the way that, for the most part, altering the way that genes are expressed. So we can alter, the two kind of themes, right, are altering the enhancer so that the gene is expressed in different locations, and that gives you different developmental outcomes, or altering the protein itself so it works slightly differently. We saw that with UBX, right? That polyalanine stretch in the UBX allows it to inhibit distillus, and when it inhibited distillus, it got rid of abdominal appendages in insects, right? So, those are the two general mechanisms. And then some of the examples of Evo-Devo that we looked at were evolution of losses. How did we lose certain structures? So snakes losing their legs, cavefish losing their eyes, sticklebacks losing their spines, things like that. Nice examples of this kind of thing. But also, obviously, there has been evolutionary gain. The gain, of course, would be our hands using that phase 3 Hox expression to specify the termini of our limbs to give us hands, basically. What else? I think that's the main stuff. All right. That's kind of the summary. Any specific questions you want to go over?
Is there a diagram?
No diagrams to label, no. No. I'm not going to put any funky Hox thing on there and say, this is C6, this is C8. No, I'm not going to do anything like that. So no, I don't think there'll be any diagrams to label like there were on the earlier exams.
There will be no technique questions this time? You know how that's...
I don't think so, no. I don't think I would ask any. I might ask about, if you do this experiment, what outcome would you expect,
Right.
That kind of thing, but not how do you do this so much, I would more likely I would give you an experimental setup. Say, suppose you did this experiment, what result would you expect? That would be more of the kind of thing I would ask. Oh, so you all remember, of course, it's Monday at 10:40. So just to remind you, it's right at the beginning. For better or for worse, we get it out of the way early. I don't like this system, but when I was in college, we had Monday and Tuesday were off. So they were basically study days, which really meant we partied all weekend and then started studying Monday instead of studying Saturday through Tuesday. But, But it is nice to have that little gap between, and that actually is helpful, right? You have that little time, you blow off some steam, and you say, all right, get to it, one last push to get through it, and you're done. So it was good. But we also had exams over a longer period of time. Our exams are crammed into literally 3.5 days. We used to have exams over the course of 5, maybe 6 days. So we would start on Wednesday. So we'd have Wednesday, Thursday, Friday, Saturday, and then Monday, Tuesday. In some cases, and really spread it out, which has its pluses and minuses. It gives you more time, but it also means you rarely had more than one exam on the same day. So it's kind of a trade-off. But the way it goes, unfortunately. All right. Well, if any other questions, I'm happy to answer them. Send me an email if you need to have